So a couple of important things. You'll notice this isn't everybody, but that's because I actually graded some of them. But remember, you need to do the agreement assignment, right? And I also pulled it up this way because I just want to point out to you guys, right? When you go into the main page for our class, right, it has a to-do list for you. For me, it's what I have to grade. But if I go to your guys' student view, it's what you need to actually do, right? So notice me as a student didn't do it yet either, so hence it's showing up on my to-do list. Right? So if you are one of those ones that it's on your to-do list, what was the deadline? Yeah, that's today, y'all, right now at 9:30. The good news is that I don't have assignments usually set up to shut down, because mostly, like, if stuff happens for you guys, which, you know, life happens, I don't have to go in and change my settings. I just go, please just do the assignment. So what am I going to say to you right now? Whatever reason you want to give me why you haven't gotten it done to this point, it's not that I don't want to hear it. I don't have time to hear it. If you need me to hear it, you can come talk to me during office hours. But just get it done, right? And I know there's the complication of that it has to be done in the lockdown browser. But that's also confirmation for me that I know you know how to access, right, lockdown browser, and you've reviewed your exam 1. Because at this point, you should have at least reviewed your exam 1. So if you had, you could have very easily then completed this very quick assignment while you're still in the lockdown browser. So go somewhere or download the lockdown browser ASAP, look over your exam, answer these few questions in my agreement assignment, and get it submitted. Some of you guys don't have a grade in the gradebook yet. And that's because apparently I asked even though it's worth 0 points, I asked a short answer question, so I have to physically go in and put the 0 in before it'll give you your 5 points for that assignment. And again, notice it's worth 5 points. So the assignment itself isn't 0, just that one question is 0. So, make sense? Okay, so get her done. Okay, so the other thing is especially after test time, students are like, can we have bonus? And I say, yeah. What does that title say? It says bonus. I already have bonus assignments in Connect. What do you have to do to get the bonus? Do the assignment. So this is our class, y'all. 6 people are sitting pretty. One person started it and didn't finish it yet. Probably said who, because I put my cursor over it. I won't do that for the others. But the rest of you guys are in the not started category. So that's my answer to bonus, by the way. It's already there. You had one for viruses too, right? Do I need to look at that one? See, virus says bonus. Right? So you don't have to do it, but if you need or want more points, then you do. So that one looks a little better. We got 13 happy people, right? How many times can you do these assignments? Unlimited. Forever. No, not really. This course does eventually end. What am I going to say if you ask me if you can do it? Do it. Okay? Just do it. Okay. None of my deadlines are, like, shut you off, right? As far as assignments go. As far as testing goes, right, you've got to take it within a certain time period. So if you miss a test, contact me immediately, right, and make arrangements to make it up. Because after a certain point, I'm going to say no to that. Because remember, I can't make the test available until everybody's taken the test, or I've said to that person, no, you're not taking it, it's too late. Make sense? But if you fall into that category and I told you no, trust me, that wasn't the only thing I said to you, right? So again, don't come to me, right? Don't take zeros. Come to me, talk to me. I have other things that I can do, right? On a situation by situation basis. Okay? The most important thing is communication. Okay, so that brings us to today's lecture. And so this one slide I added as a PowerPoint because I thought I had it in my PowerPoint, and apparently I didn't. It was in my other class, and I realized it would probably benefit you guys too. So under, and I just added it, so you'll see gene expression. This is literally just one PowerPoint slide that I've now added to my PowerPoint. And so we're going to go ahead and take a look at that. So this one basically describes this table, right? This is the code, right? The translation code. There are 64 codons, right? Remember that codon is the 3 nucleotides, 3 letters together. And the ribosome is going to read that. And if it is a sense codon, which 61 of those are sense codons, it's going to put a specific amino acid there. 3 of the codons are called nonsense codons because they stop translation, right? They're what we also call stop codons. But the terminology is also a nonsense. And this will make sense because, remember, do we have nonsense mutations? Yeah. Yeah, and what happens? What gets put there? What's nonsense codons? What do? They tell the ribosome to stop, to terminate. So do you see how looking back, I was like, oh, this might help you guys, right? Right? So we're not by any means memorizing this code, right? But we need to understand how it is utilized and why changes sometimes will result in changes in the amino acid, in the phenotype, and why sometimes we will have changes in the genotype, in this code, and not have a change in the phenotype. The amino acid stays the same. So let's review a little bit. I actually augmented one of my slides too so I could quiz you guys this morning. Should have put it first though. All right. So this is how it's supposed to be. It's supposed to be tyrosine, right? The DNA and the RNA, it's all supposed to code for this amino acid tyrosine. Over here, this is the original code. Here it's changed, right? And so has the codon, right? That's going to be read by the ribosome. But the amino acid has not changed. So it's still a change in the DNA. So it's still a mutation, a change in the genotype. But no change in the amino acid, no change in the phenotype. What do we call this type of mutation? This is a silent mutation. Now this next one, again, it's a mutation, right? A change in the DNA, change in the codon, change in the amino acid. So we've got a phenotypic change here now. Right? The physical expression has changed. What is the name for this type of mutation? Missense. Right? Because remember, sense codons code for amino acids. This makes sense to the ribosome, right? It codes for an amino acid. The only problem is it the correct one? No. So it's the missense mutation. Right? You can think of it as a mistake, right? The next is, this actually codes for a stop codon. And what do we call stop codons? We just learned it today. Nonsense. So what type of mutation is this? A nonsense mutation. So maybe helped a little bit? Right? Having that one additional slide with some terminology. Okay. And then, of course, what we call something in its natural state, where it hasn't mutated, is the wild type. And I think that's where we ended up last time, right? Good. I didn't even cheat and look at my recording. my memory served me right. So we're looking at just a single base changes. Right there, those were point mutations that led to possibly a silent mutation, a missense, a nonsense mutation. Next, we're going to look at the consequences of adding or removing a single nucleotide, right? So a single base. So in this case, again, this is the wild type. This is the DNA. We know that because we see T's, right? And this is the messenger RNA because we see the U's. And then the ribosome is going to read it in groups of 3 of these codons, and it's going to put the corresponding amino acids in place to form the eventual protein. If for some reason an extra base is inserted, because of how the code is read, wherever it is inserted, it shifts everything downstream. So we call this phenomenon a frame shift, because the reading frame is going to get shifted. Because we added one right here, it doesn't just affect this one codon, it affects everything downstream from it. Because it just shifted everything. Because the ribosome doesn't know this happened. It's going to stick to its usual routine. 3 nucleotides, codon, amino acid, codon, amino acid, codon, amino acid. The only problem is we just screwed up the code. Look, right? By adding that one T right there, we've shifted it. So now it's reading a totally different codon. It put a totally different amino acid there. Same thing in the next position and in the next position. And this is bad news too, right? Because it's not even an amino acid, is it? It's telling the ribosome to stop. So the most common result of adding or removing and shifting that reading frame is what we call a premature stop codon. Stopping before you're supposed to. Remember, the consequences of stopping before you're supposed to, how could that affect a cell? Is that good news? Bad news?
It depends.
It depends on how important that protein is, right? So if that's a pretty important protein, this is what? Definitely bad news, right? If it's a protein you could survive without, you're probably at a disadvantage, right? But the good news is you're still alive. So it's going to vary, but most of the time, really not good news. So in the bonus, right, are the animations. Then I also have links to them to make it quick and easy for you guys without having to even use the ebook. Speaking of which, I asked somebody that question. For you guys, you could buy Connect by itself, right, for like $23 or something in Connect. Do you guys not have the ebook anymore? Or do you? Or have you even checked? Nobody knows because everyone bought the paper version. You don't? Okay, so you definitely don't. Yeah. Right, okay. So that's a glitch in their system this semester, so you guys totally made out, though, by the way. You weren't supposed to be able to buy the $15 copy unless you purchased the ebook. So, yeah. You guys rocked it big time. So, I know you're happy about that. I'm happy for you guys, too, because my students have been robbed forever. So, what did I want to show you guys? Oh, so under the module, right? And if you download my PowerPoints to your computer, in order to play the animations embedded in the PowerPoint, you have to go into presentation mode like I do when I present to you guys, and then the buttons work. But then your other option, too, is just going over to our Module 2 animations and videos, and I have the links, right? They'll go to the web page within the free portion of the companion site for our textbook. So these will open up. So let me just open one just to be sure. I can't remember why I test run in this semester. Yeah, so the only thing is it's kind of small. But, you know, if you're on your individual computer, then it's not a big deal. So I'm going to stick to my PowerPoint, though. And I am going to go into presentation mode, right, so my buttons will work. And I'm actually going to turn off the audio. But notice, too, they have a transcript as well of what they say. But I'm, of course, going to add my own soundtrack. Right, so I'm just going to pirate their animation. So here we have the wild type, how it's supposed to be. Of course, it's DNA because it's double-stranded and we see T's. So in this one, and then it shows you the messenger RNA and then the amino acids that message would code for. So here the cell maybe made a copy and for some reason put an additional nucleotide in here. So that just messed up, right, the reading frame from this point down. And again, just like the other example we saw, it messed up the codon where the change happened, where the addition happened in this case, and the one next to it. And again, the worst case scenario happened where they stopped way before it was supposed to. So that's adding, but what would happen if we took one away? Yeah, are we still going to get a frame shift if we take something away? Yeah, we're still going to shift it, right? Because, again, that ribosome's like 3 nucleotides, codon, 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 right? That's how it works. It's not going to change its system, right? Because you screwed it up, you know, further down the assembly line. So this one shows you, right, where they deleted one. Same problem, right? Where the change happened on down is affected, right? It shifted everything from that point forward. In this case, you know, they're not showing necessarily the whole gene. I mean, we're just looking at 4 amino acids. This would go much longer, right? And probably at some point they're stopping before they're supposed to. So, other than my poor grammar, right? There was a point I was trying to make here. The cat ate the dog. These are all 3-letter words, are they not? So, you know that same system that the ribosome uses, right? 3 letters is a codon. In this case, 3 letters is a word in the English language. Again, grammar's bad, but you can read that, right? It somewhat makes sense, other than the grammar not being on point. What'd I do? I shifted it because I did what? I took out the T, and I did just like a ribosome would do. I read it by groups of 3 letters was going to be a word, right? So it becomes heck 8 at, yeah. This is not even Pig Latin. This is like totally destroyed English language, right? Gibberish. Right? So, the same thing, right? Nucleotide, the ribosome doesn't realize you have it reading gibberish, right? It just reads, right? So if you were to give some kid this, like I just did, they're going to try and read this, right? Based on what they know. They don't realize you screwed it up. Or as far as my son is concerned, he thinks the copy machine messes everything up. Did I tell you guys that already? When there's something wrong in his homework, he says the copy machine made the mistake. So I've literally brought him here and showed him that a copy machine literally just makes a copy. The original had the mistake, not the machine. It is not the copy machine's fault. And again, it's not the ribosome's fault, right? It just reads based on its system, right? So what did I do to this one? I added the A. So the same problem though, right? Because I stuck to the system. 3 letters, it's a word. No, it's really not a word, is it? We got one word. We got lucky, right? We got T. Other than that, this means nothing to us. So, you know, I'm using whatever you call it. An example. Can't think of the correct term. Analogy, thank you. Using an analogy here. So I could put the word big in, right? And remember, what's the analogy here? So the words here represent what in DNA, RNA, language, and ribosomes and all that jazz? Codons, all right? They're codons, right? So if I add a codon, essentially, by adding the word big, where could I add it? Would help us out a little bit. Yeah, maybe the big cat ate the dog. But I didn't change the sentence, right? We can still read it. We just did, right? Again, the grammar is not there, but you know.
You just described that cat.
Right. This in DNA language, as we said, is a codon. So what's the ribosome going to add? An amino acid. What didn't happen that happened before when we added just one? No frame shift. No frame shift. So this is the one very small exception to adding or removing. You have to add or remove an entire codon in between codons in order to not get a shift. The likelihood of this happening, very small. And again, what would the consequences of that be? We just added an amino acid, right? We got this protein, let's plug in an extra amino acid. No frame shift happened, right? We just put in an extra one. Yeah, just plugged in an extra one. Depends on how important that one amino acid is to that protein, right? Could make that protein super awesome. Could happen. Could also make it non-functional. Could have no effect at all. It could really run the gamut of possibilities. Same is true if you removed an entire amino acid, right? Could be really bad, could be really good. Could have no effect at all. Change really is determined by, right, what does it do? And really, what environment does it live in? Right? Environment puts a huge pressure on whether change is going to be successful or not. So this is in DNA, RNA terms, right? And amino acids. So this is our wild type, right? This is how it's supposed to be. I made that change. What did I change? What did I do? I added a codon. Which codon? CAC, right here. So that essentially added what? An amino acid. But did I affect the code? Did I shift it? No, right? So notice it's the same. It is kind of shifted over a spot, right? But it isn't shifted as in we've messed up the reading frame. So who discovered transposons? I don't know. So this is a guy you need to know. Or girl. Oh my God. Shame on me. Forgot. This one's a woman. There are usually so many guys, right? I forget. We've made significant contributions as well. Barbara McClintock. And I think, unfortunately, she's dead now. Oh, yeah, probably one more A right there. Yep. Hopefully she won't come after me. Yeah, most of the time it's the last name that's important, right? So Miss McClintock. And these transposable elements, or transposons, are sometimes affectionately referred to as jumping genes, are segments of DNA. So it's DNA that spontaneously move from gene to gene. So what is a gene? Redefine that? So when we say gene, we're talking about a section of DNA. But in that case, we're talking about a section that codes for something, usually makes something, right? So, when your RNA polymerase makes messenger RNA, it reads only a particular section of that DNA. That section is referred to as a gene. It codes for something, right? That message is made, the ribosome attaches, and it makes that protein. Right? Hemoglobin, that protein in your red blood cells. You have a gene, a segment of your DNA dedicated to making that protein. But as we just saw, keeping that information intact is very important, right? We need it all together. We need the messenger RNA to read it all together, make that transcript, that message. We need the ribosome to read the whole thing and make that protein. If we take another piece of DNA, this transposable elements, and we insert it into that other one, it's going to disrupt that code, right? It's going to separate it. Our enzymes, remember, slide down that sugar phosphate backbone. They don't jump, right? So are they going to be able to put those two pieces back together? No. Instead, they're probably just going to copy that transposable element, that transposon, whatever that is. But whatever it jumped into and disrupted has now essentially been knocked out, or sometimes referred to as insert mutation has undergone. So this happens in nature. It's actually why, there we go, there she is. I got her A on there. There's Barbara. She was studying maize corn. So those of you guys that don't know what maize corn is, this is a picture of it. It's the corn where you'll see, and sometimes it's referred to as Indian corn. This is one of the more natural strains of corn that existed before we started screwing with it. So genetically, each one of those kernels is a baby plant, right? That would give rise, that's the seed for another plant. Each one of the individual kernels is an individual seed. These are all her babies, basically, for that plant. So each one of them, though, is genetically similar, because plants aren't quite do the stuff like we do. But anyways, why do we get all these different colors? It has to do with the DNA, right? But actually, it has to do more with the expression of the DNA. What's being expressed and what's not being expressed. So when she delved in further, she found out that even though the gene was there, in some cases it was disrupted by other genes. And that's how you ended up with these different colors. So even though the information was there, it was disrupted, so it couldn't even be expressed even if it wanted to be. And so in this picture here, this is showing you two circular pieces of DNA, what we commonly refer to as plasmids. So here, the numbers represent individual genes, right? Number 1 codes for a particular protein. Number 2 codes for something else. 3 codes for something else. So, I mean, it doesn't even have to be just one gene. Several genes together can move, can jump. Yeah, both of these circles represent circular pieces of DNA, commonly referred to as plasmids, right? We find these in bacteria a lot, right? So this is just an easy way of representing it, being circular like this. And scientists work with circular plasmids a lot because they're really easy to work with. So even though this phenomenon happens in nature, we've figured out how it happens, and we can actually cause it to happen in laboratory situations. So here we have another plasmid with the genes A, B, and C. So again, A codes for something, B codes for something, C codes for something. This jumping gene segment, these jumping genes, jumped into B and notice disrupted it into 2 pieces. Part of B is here and part of B is over here. The problem with that is basically B is non-functional now because those 2 pieces have been separated. They can use 1, 2, and 3, right? They're not disrupted. That transposable element itself is usable. The gene it jumped into, though, and disrupted, is now non-functional. It cannot be made. So, as I said, scientists sometimes do this on purpose, right? Yeah, it would be one step in the cloning process, potentially. Okay, so I'm going to give you a real scientific example of similar to stuff I did in the lab when I was in grad school. So we have strains of bacteria. Again, we're dealing with plasmids, so these are actually circular pieces of DNA, but I've just stretched it out just for ease of representation. So again, there is information there within the DNA code that tells the enzyme start here and stop here. And so it would start and it would copy and it would make this protein. In the case this protein that this line of bacteria have, code for an enzyme that can split a substrate that we give them that's colorless when we give it to them. But if they produce this enzyme, they will split it and it will produce a blue color. It's really kind of cool. So we can tell, right, if genetically they have this enzyme because they will be able to do this action. They'll be able to turn blue. E. coli doesn't usually grow blue, right? But in the presence of this substrate, they'll metabolize it and they'll produce this blue color. If we take another gene and we insert it into this gene, are they going to still be able to do that? No. But what are they going to be able to do? Whatever this is that I gave them, right? Whatever this blue piece is, it's intact, right? In this case, I just gave them antibiotic resistance. Whew. Why would I want to do that? So I can select for ones that have genetically changed. Right? Because just because, right, they can't turn blue doesn't necessarily mean, right, that they don't have that enzyme in their DNA. They might just not be using it, right? Something might not be turning it on. I want to know when I give them, so this is usually a selective marker that we give them. So we don't just give them penicillin, right? This one wouldn't work on E. coli, right? But you guys know what penicillin is, right? It's an antibiotic. Right? So if I give them antibiotic resistance, I give them something else. So in the case of the research that I was doing, I was actually literally giving them a huge piece of DNA from a frog that I had amplified, right? And I wanted to sequence that, but I needed to be able to have lots of it to be able to run it on the sequencer, to find out the actual code for that particular gene I was looking at in the frogs. So what we would do is put them in, we would take those pieces of DNA and we would put them in this construct in a plasmid, right? And then insert them into the cells. Okay? So we would stick pieces of DNA into pieces of DNA, into a circular piece of DNA. And we give it to the bacteria because guess what? They're really cheap copy machines. Really freaking cheap. And really pretty good at copying, right? Not likely to make a mistake. So they would copy that plasmid for us over and over again. Then we just bust open their cells and steal the plasmid back. But that's not a guaranteed process, right? I wanted to make sure that they took the DNA I needed them to take and copied it. How was I sure of that? Because if I grew them in the presence of the antibiotic, it would normally kill them. But because I gave them antibiotic resistance, they'll survive. So I could very easily pick and know only the bacteria growing on that plate that were white, right? Because again, I disrupted the ability to turn blue. They're growing in the presence of the antibiotic only because I gave them that resistance, along with the DNA I wanted them to copy for me. Pretty cool, huh? So the one thing I don't like about this animation is the level of detail. Right? So it's really cool, right? So here's a transposon, right? This is a segment of DNA that's going to move. Here's another one, right? So what do? They show it moving. Going from here to there, and there to there, and even back onto a plasmid and leaving. Oh, that's all well and fine. But what is happening when this gets stuck in here? It's disrupting the information that was there, right? Whatever was there just got knocked out, just became non-functional, right? So although, cute animation, I don't know that it really helps us understand the level of detail we need to know, right? We're getting disruption happening here. Right? When those guys jump, right, whatever they jump into, they destroy. Not permanently, though, because as you notice by that animation, they can jump back someplace else. Right? They can jump around. Now I got that song stuck in my head. What was, crap. Something else I wanted to say. It just escaped me. Oh, I remembered. So, when I was at LSU doing research, at LSU Vet School, there's an immunologist there, who of course the name is going to escape me right now. Really cool woman. Like, frighteningly intelligent woman, actually, working in immunology. And she had a whole line of mice that had a particular knockout mutation, right? Where the mouse at the organism level could not do something so that she could study its effect on the immune system. Right? So, pretty cool stuff. Like, when I was just out of undergrad, right, working in the animal unit, and they're like, oh yeah, those are such and such knockout mice. And I'm like, what the hell are knockout mice, right? What are they talking about? And then somebody explained to me one day, I was like, wow, that's crazy, right? Pretty crazy what they can do to be able to study. Okay, so mutations are essential for studying genetics, right? What if you can't do this? What effect is that going to have, right? What if you can do this? So, although they can be intentionally produced, right? We can cause mutations to happen to demonstrate function of a gene or set of genes. When we do that, we refer to those types of mutations of being induced, right? We purposely caused it to happen. Now, so transposition, right? Transposons, pretty widely used in genetic research, which then, of course, can be applied to other disciplines like immunology. Chemical mutagens, not so much, right? We found out these chemicals mutate and change your DNA, right? We don't necessarily use them because they mutate your DNA, right? We don't necessarily use chemicals. Although in some experiments, depending, right? Most of the time it's, we found this chemical, we want to make sure, right, that it doesn't cause mutations in DNA, because that could be bad. And then radiation, we've known for a long time, right, causes distortions in the DNA. And if you damage it enough, you can cause death of the organism. So as far as chemical mutagens go, there's tons of different classes of mutagens. I'm just going to mention one particular class, and it's called intercalating agents. And ethinium bromide falls into this classification, and one of the reasons why I picked this one is that this is very commonly used in laboratory research. Again, not to mutate DNA, but to help us visualize DNA. Because what this chemical does is it binds to DNA, right? It intercalates, it inserts itself into the strands of DNA. The cool thing about that is that when you shine UV light on it, on this chemical, it fluoresces. So these lines that you see, right, when they show you guys gels, they've taken DNA and they put in this gel matrix and they applied an electrical current, and because DNA is negatively charged, it's going to head towards the positive pole. And that speed and how far it's going to go is directly related to its size. Because this gel matrix is just that. It has little tiny microscopic holes in it. And so really big pieces of DNA aren't going to travel very far. They're going to get hung up and stuck. So you'll see this guy at the top. I know that's a pretty big piece of DNA. And I'm not talking about a single piece of DNA, y'all. I'm talking about copies upon copies, like thousands of pieces that are all the same size. Where down here, this band is, thousands of copies of a much smaller size. Because see how it went further down? That sample. And over here, you see a whole bunch of lines, right? This is commonly referred to as a ladder because it kind of looks like that, right? But this is the known or standard. So they know what the size of each one of these bands is. How many base pairs. So they can kind of, estimate, well, my sample, my unknown sample, right, where it lines up, we know about how big it is. But the reason why we can see these guys is because they've stained this gel or the sample contained ethinium bromide. And it's binding to the DNA. So when we shine the UV light on it, it fluoresces where only the DNA is because it binds specifically just to the DNA. This bottle, if you were to pick it up in the lab, has a skull and crossbones on it. It says mutagen carcinogen even. Because as you can imagine, right, it inserts into the DNA. So if you were to get it in your DNA, what is it going to do? Same thing. It's going to insert. The only problem is you're going to be actively copying your DNA, yeah? Right? So what happens when you've got this extra space here? When it's copying, what is it going to probably do? No. Think about this process, right, of copying DNA. You've got this big old space. right? You tell a carpenter, right? Make me a set of stairs. You don't want them, you know, all 1 foot apart and then some big 4-foot gap, do you? I don't know about you. Stairs are hard enough. Never mind having to jump up 4 feet. Remember how structurally important DNA is? It's uniform. It's that beautiful spiral staircase. It's not going to have a 4-foot gap, right? So the enzymes are going to do what? They're going to add a step. They're going to add a nucleotide, right? What happens when we add nucleotides? Just learned it today. You get a frame shift, and typically you're going to do what before you're supposed to? You're going to stop. Do you want that happening to your DNA? Yeah, I don't think so. Right? So I always wore gloves. I always protected myself. One of my colleagues never did. He also smoked like a fiend. Obviously doesn't care about his DNA. The way he looked at it too is like your skin cells are mostly dead on the surface, right? But you know, some of that could penetrate deeper. And I personally don't want skin cancer, so I kind of avoided contact with that substance.
Like when we stain microorganisms, they die.
Mm-hmm.
What happens to the DNA when it's stained? Does it?
Which DNA are you talking about? Our DNA or the DNA that's in it, basically a test tube and now in a gel?
That,
Right. So,
moving, then it's
right.
not living, so I'm not-
No, it's not moving. It's being attracted, right? DNA is a molecule, right? And it's mostly negatively charged because of the phosphate groups. So when we put in that gel, we apply an electrical current. It moves towards it. You know, opposites attract. It moves towards that positive anode because it's negatively charged. So that's why it's moving. It's not moving on its own. We're using electricity to move it. Right? We're using charges. Yeah. And you wouldn't take this, say, out of this gel and go stick it in an organism. Because, again, you can't unstick the aethanium bromide to make it functional anymore. So you would have taken a sample out of your original in order to run this gel. And so then you would have your original unstained stuff, right, that you could then do experiments with that would be safe for an organism. Good question, though. I like the thinking. Okay. Radiation. There's 2 types of radiation. Ultraviolet radiation and X-rays. Ultraviolet radiation causes what you see in the diagram below. When ultraviolet light shines on 2 thymines that are next to each other, instead of hydrogen bonding like they're supposed to the adenines on the other side of the double strand of DNA, which is a relatively weak bond, which is why we can separate the pieces of DNA to be able to, you know, replicate DNA and make transcripts of it. When UV light shines on it, instead, 2 thymines next to each other covalently bond with each other. What kind of bonding is that? My chemistry people should know, right? 2 nonmetals. What about, what makes up an atom? They're negatively charged, and some of them are positively charged, but I'm more concerned with the negative charges going on. Those things that spin around the neutrons and the protons. The electrons, right? The electrons are actually being shared between the 2 thymines. That's a much stronger bond than hydrogen bonding, right? They're like seriously holding hands. Red Rover, send Julie over, right? You ain't breaking through, are you? You're not breaking through. But the problem here isn't necessarily the bonding of the 2 thymines. It's the effect that that has. 1, it's a strong bond, and 2, what happened to the backbone of the sugar and the phosphate? It's bulging out, right? So this is the bump on our banister, right? So imagine that sugar phosphate backbone, that railing of the banister to that beautiful spiral staircase is got a bump in it. So what happens to the kid who tries to slide down? Why do we put bumps? To make them fall off or stop doing it, right? Do we want our enzymes falling off? No, this is not good, right? We need smooth, beautiful banister. This is not smooth or beautiful. This is a problem, right? This just made that section of DNA unreadable by our enzymes, by our RNA polymerase or DNA polymerase. That's our code, that's our information. Is it good that we can't read it now? No, that's bad news. So, are we exposed to UV light? All the time. Are we exposed to too much of it? Sometimes. Depends on what you do, right? Stupid me didn't put any sunscreen on for my son's soccer game this weekend. I got burnt. I was like, oh yeah. Not doing that again, right? How many of you guys know Dean Gruber? Might be a few of you guys. The dean for math and science. Old guy, sits in the office. All right. If you ever pass by, right, his nose is not beautiful anymore. I'll love him to death. But maybe 2 surgeries now. Same thing happened to my dad. Coached baseball. Stood outside in the sun all the time. Ended up with what? Skin cancer on their noses. Right? Didn't protect themselves. Overexposed themselves, per se. Right? To radiation. Tanning beds. What's the lights in those? UV lights. Not a good idea, y'all. Purposely overexposing yourself to something that does this to your DNA. What's the good news? Because you're like, oh, crap. Right? No one wants to go outside right now. And certainly not jumping in a tanning bed anytime soon. Is this permanent? No. No. The good news is we do have enzymes that can reverse this and protect us. But you don't want to overburden the system, right? Because if you overburden the system, at some point it's going to miss it, it's not going to fix it, and it's going to create serious problems. X-rays. This is worse than UV light. It literally shoots electrons at you. Yeah, and guess what it's doing to your DNA? Chopping it up. Chopping it up. Can we fix this? Yeah. We can. We can. We can. It's harder if it chops both sides, right? But if it just nicks one side or the other, we have enzymes, we can fix that up. Right? But this is why, what do they cover up if they can? No. Yeah, because what's in your abdomen? For women, it's inside. For guys, it's outside. Your reproductive system. Because those cells, right, if they get damaged, right, and don't get repaired, you pass that on to your children, right? That distortion, right, that damage, right? So that's why they're always going to protect, and that's why if you're pregnant, they're not going to do it at all, right, unless they absolutely have to. Which actually I've never heard of a case where they absolutely have to. They're going to do other things. Huh? Yeah, magnetic. That's what I'm saying. They're going to do something else. We have other things other than x-rays now. And this is also why you're not going to go get x-rayed every day. You know, it's not fun for your body. This is why the technicians that do it leave the room, right, if they can, or they shield themselves. Right? And so the same is true when you go to the dentist. That's why they put that big lead vest on you, right, is to keep that from penetrating. And what they're really trying to protect is your sexual organs more than anything. Make sense? So, do we use this on purpose to kill things? Sometimes, right? We use UV light in laboratory settings to keep surfaces clean by overexposing microbes to UV light, so much so that they can't repair, that they die. We've even used it now for helping disinfect water, microbes that are in the water. The problem with that is it doesn't penetrate very far. And that's why you're like, oh, how is it that sun cream works, right? We're just putting this layer of goo on us, right? Some of it's kind of gooey. This very thin film, right, can help protect us because UV light isn't very penetrating, right? And so that's why sunscreen works. It helps keep it from penetrating. Something as simple as saran wrap. You really can't get burnt sitting in your car with the windows up. You'll get hot, but you're not going to get burnt, right? The glass protects you for the most part. But not all glass is the same protective as others, you know. So some glass does let in to a certain extent. That's why some sunglasses and things like that, right, have UVA and UVB protection, is to really minimize it to almost nothing that gets through the glass. Okay, so enzymes will fix the problem, yay, right? So we need to be able to identify the enzymes that have the ability to proofread DNA as it's being replicated. So this is actually pretty easy. Guess what? What makes your DNA? DNA polymerase. Can it check its own work? Yes, it can. Yes, it can. Just like you can check your own work, right? So does the same thing that makes DNA, it also proofreads it as it's making it. And it can fix its own mistakes. So there's different types of repair mechanisms. So excision repair, direct, mismatch, and SOS. Right? What does SOS stand for?
Save our ship.
Save our ship, right? So this is the last ditch effort to save themselves, right? When SOS repair is online, they're like, oh, our ship is going down. This is an emergency. So, and again, remember this is microbiology class, so we're focusing on bacteria, right, to repair their mutated DNA. But some of this stuff happens for us too, right? So we need to, in addition to how these systems work, and we're going to keep it simplistic, excision repair, what is that fixing? What type of mutations that we've talked about will excision repair fix? What gets fixed by direct repair? What gets fixed by mismatch repair? And because of that, too, remember, if you look, when the DNA is being copied, remember, we split it and we use each side as a template for the new strand. So the cell can actually tell the difference between the original template strand and the new strand. And that way it knows where the mistake was made, right? You don't need to fix the template, you need to fix the new strand where the mistake was made. If that doesn't get fixed, though, then that becomes your template strand, right? And now you have a mutation. So DNA polymerases I mentioned already, and there are actually lots of DNA polymerases. But in general, they have the ability to excise, remove the incorrect stuff and replace it with correct stuff as they're making the DNA. Excision repair corrects damage that causes distortions in the double helix. So what type of mutation did we just talk about that will create a distortion in the double helix? The UV light. The thymine dimers, right? Those two thymines binding with each other that form when UV light exposure happens. So how are we going to fix that? So see the distortion right here, right? They're covalently bonding with each other. They're super best friends, right? We're going to literally, excision means to cut. We're going to cut out that section and we're going to replace it. So some of our nucleotides too can get converted into the wrong ones. So notice here, this is DNA, right? This is double-stranded. We're seeing mostly T's. What the hell is that U doing there? It's not supposed to be there.
Pardon?
Right? Something happened. It's been changed. So again, we have special enzymes that are going to detect this, and they're going to fix these distortions. Direct repair is another way we can fix things like the distortion caused by UV light and causing these thymine dimers. But the difference here is we're not going to cut out a section. We're going to directly repair it on site. We're not going to remove anything. And sometimes this is referred to as light repair or photoreactive repair, because the enzymes that actually break apart these two thymines that stop this covalent bonding use light energy to do it. And guanines sometimes will get damaged too, and there's a type of direct repair where the enzymes directly attach, right? They're going to use energy, because you need energy here. You know, Red Rover, send Susie right over. You need some serious force to break through this serious hand-holding going on here. So in contrast, sometimes the excision repair is referred to as dark repair because you don't need light. These enzymes don't rely on light to cut out these sections and replace them. So for some microorganisms, we can expose them to UV light, and if you keep them in the dark, they can't repair themselves because they rely on this direct repair that uses enzymes that use the power of light to break that bond. But if they have the excision repair, right, they don't need light for that. They're still going to fix themselves. Mismatch repair, as the name implies, this is when you have mismatches, right, where you've got a base substitution going on. And this is really important. So I already answered the first question. What do we call this when it's the wrong amino acid? I mean, nucleotide. A base substitution, right? They put the wrong one there. What's supposed to be there? C, right? Now I want you to look closely. This is labeled as the template strand. This is the new strand, of course, where the mistake is. What's the difference between this strand and this strand? What's this? Ah, methyl groups. Chemistry people. It's a methyl group. When your DNA, as it matures, it gets what we call methylated. And this is so your body knows this is the mature template strand of the DNA. The new stuff, right, it's brand new, so it hasn't been methylated yet. It hasn't been checked out, right? It hasn't passed inspection. It has not gotten its inspection sticker yet. So the enzymes know, oh, this is where the mistake is. Not on this side. Here is the mistake. So very similar to excision repair. It's just going to cut out that section. Because this is, notice y'all, their nucleotides at DNA polymerase already went by. He didn't catch his mistake, right? He's on downstream, trucking along, thinking he did everything perfect. Not the case, right? Other enzymes are coming in behind him going, oh no. You goofed. I found it. Don't worry, I'll fix it. I'm going to cut it out. Your friend is going to come by and put this section back in. We want to paste it all back together again. And we're good. So how do we tell the difference between old and new? The old has been methylated, right? This CH3 is called a methyl group. And that's important, right? Because we want to make sure we fix the right strand. We want to fix where the true mistake is. So when DNA damage is very severe, replication will stop altogether, right? The DNA can't be copied if you've got a whole bunch of distortions and breaks and mismatch. I mean, it just overwhelms the system. So a special type of system is triggered called the SOS response. And in this case, DNA polymerases are made that can go over the bumps, can deal with the distortions. The problem is it's going to basically kind of try and fill in the gaps on its own. So it inherently will make mistakes, but it'll still keep copying. Unlike the normal polymerases are like, no. I only deal with perfect staircases. I'm not a danger jumper. It's not happening. Right? Where these other guys are like, they know it's the last ditch. Right? These enzymes will just keep going. Right? They're going to do the best they can to figure out how to put it all together. Right? If they don't make too many mistakes, that's great news, especially if you're a bacteria, because you're a single-celled organism, right? You now may be able to continue and reproduce. Or you might be done with. Because the damage is that bad, right? It's either you're done with or you get to keep going. So you're going to try everything you can to keep going. In this case, you're like, I can deal with some mistakes, right? As long as I can keep going. So mutations, these changes in the DNA, in the germline, provide an organism a way to respond to changing environments. Because if we never changed and the environment keeps changing, we're going to get wiped out. If we can't adapt, if we can't change, we will be gone. So the important thing is, y'all, although environment like UV light, we know causes change, the environment itself does not cause this change. This change happens naturally on its own. The environment, though, determines who ends up being good and what changes are bad, right? If you change, it's good, you're going to keep going, right? Because the environment's going to let you. If that change makes you not best suited to that environment, eventually you're going to be eliminated by that environmental pressure. We refer to that as what? Natural selection, survival of the fittest. So next time we're going to look at in science, how do we figure out and isolate mutants and study them, right? Because of what the consequences of mutation are, there's sometimes going to be different ways that we'll be able to get the mutant and be able to study it. And that's what we're going to look at next time. Remember, all the PowerPoints are already posted.